So today we're starting a section 3.4C, and we'll actually be talking about other types of technology that's involved in studying genetics. So we'll be on page 6870, and we're going to start by talking about some chromosomal disorders. We've already talked about genetic disorders, disorders that are caused by mutations in the DNA or just by the wrong base, nitrogen base, replacing another base that's needed. But today we're looking specifically at chromosomal disorders. And this means that the disorder is inherited due to problems with the entire chromosome. And so this is very different from our genetic disorder. Let's review a little bit what that means. Remember that a chromosome, it could be single, or it can be doubled if it's gone through DNA replication. Okay? But in a chromosomal disorder, we're saying that this whole chromosome has some sort of problem. It might be that the chromosome is broken. It might be that the chromosome is damaged. It might be that we have an extra chromosome, or it might be that we don't have this chromosome at all. How should we have our chromosomes? Well, all of our chromosomes should come in pairs. There should be two chromosomes. It might be doubled, it might be single, and it'll just depend on what phase we are in, if we're in mitosis or meiosis. But they're always going to come in pairs. Sometimes when we have a problem or a chromosomal disorder, we might actually be missing one of those pairs completely. We might actually just have one chromosome and are missing the second chromosome that should be there. Or sometimes we have the case where we might end up having 3 chromosomes total, and meaning that we have an extra chromosome. But usually the mistakes in these chromosomal disorders are mistakes that are caused in meiosis. Whereas if we had a mistake in a genetic disorder, like Huntington's or cystic fibrosis, that means that a small section of the DNA is damaged. So a small section of the DNA within the chromosome. Because remember, chromosome is just DNA that's been coiled up. When we're talking about a chromosomal disorder, normally what causes this is called non-disjunction. Non-disjunction is the process that causes the chromosomal disorder. And this is when chromosomes fail to separate properly during meiosis. And this leads to a problem known as aneuploidy. So this is the problem, this is the result. And aneuploidy means that we have an incorrect or extra or missing number of chromosomes in a fertilized zygote. So if you're looking at these two pictures, they're actually showing you steps in meiosis. And what you actually might want to do, or what you actually should do, what you should do is draw one or the other, one picture or the other, really tiny in the bottom of page 68, okay? And it should fit just one of those. Here they're showing you that you start off with 2 chromosomes. Notice that these chromosomes have been replicated, so DNA replication has occurred. DNA replication. And then here you have the chromosomes separating, okay, going through meiosis. And then here you have your chromosomes separating again. So remember that this happens 2 times. You have meiosis 1, and this is a good time for review, and you have meiosis 2. And at the end, how many chromosomes should you have in each daughter cell? Well, at the end, you should have 1 chromosome, CHRMS, in each daughter cell. And you want to add this in. You should have 1 chromosome in each daughter cell. But instead of having 1 in each daughter cell, look here, we have a problem, a mistake. And this mistake is called aneuploidy. It failed to separate, there should just be one L, failed to separate is non-disjunction. Okay, so failing to separate, when we were separating our chromosomes, is called non-disjunction. So that's the process that happened during meiosis II in this case. And the end result where we have these two cells having the wrong number of chromosomes is called aneuploidy. So make sure you take a minute to pause, relabel those things, and that way you have it in your notes to clarify. Non-disjunction is the process. We failed to separate properly. Aneuploidy is the result. Now we have the wrong number in these cells. Okay. Here again, they're just showing you that picture, and what they're really showing you is that the mistake can happen during meiosis I, or it can happen during meiosis II. So it can happen the first time it separates, it can happen the second time it separates. Now, we have three disorders that we need to know about that are caused by non-disjunction. And the first one is Down syndrome. It's a pretty common one. What we have here in the picture is, and please take a minute to pause the video, copy those notes first, and then hear the explanation about how do we figure out that someone has Down syndrome. So Down syndrome is caused by an extra chromosome, number 21, and we also call that trisomy 21. Trisomy meaning 3 chromosomes. Okay, somy, chromosome. So 3 chromosomes at location 21. What does that mean? Well, here we have a picture known as a karyotype. And in this picture, what you see are the chromosome pairs. And all of the chromosome pairs have been labeled or numbered from number 1, pair 1, all the way to pair 22. And then at the very end, pair 23 are your sex chromosomes. Okay? So you actually don't have this exact picture, so we'll label this again in your next picture. But for Down syndrome, what you see is that on the 21st pair, trisomy 21, you have 3 chromosomes. How many chromosomes should you have to make a pair? Well, you should only have 2. So for each of these, you see 2 chromosomes. But for pair 21, you actually see 3 chromosomes. And having that extra chromosome is what causes these problems. And there's actually a lot more problems associated with this disorder. What we'll need to be able to do is be able to read these karyotypes and pick out where's the problem. You might see 1 chromosome missing, so instead of having 2 to make a pair, you just have 1. You might see an extra, and this can happen anywhere from 1 to 22, those are called your autosomes, or on the 23rd pair, your sex chromosomes. Here's where we have our 2 other chromosomal disorders. We have Klinefelter's and Turner syndrome. In Klinefelter's, the male has an extra X chromosome, so this is your figure. This is your figure 3.4i. And on 3.4i, you want to put a box around those 2 X chromosomes and your Y chromosome. The other thing I want you to do is label next to the picture is that chromosome number, numbers 1 through 22 are called autosomes. And we've seen that word before, autosomal. And then pair number 23 equals sex chromosome. That determines your gender. So pair number 23 equals sex chromosomes. Chromosome numbers 1 through 2 equals your autosomes. So here we're having issues with our sex chromosomes. Same thing for Turner syndrome. Turner syndrome means you have a female with only one X chromosome. And when we write that down, we'll actually write it as XO instead of XX. So not XX. They're actually missing an X chromosome. And please circle that, or actually I think that's already sectioned off in your picture. And then, now we have some diagnostic tests that can be done. Detection of disorders. There are some tests that we can run to detect different genetic disorders. This could be the genetic disorders. This could possibly be the chromosomal disorders. For the chromosomal disorders that we just looked at, Down syndrome, Klinefelter's, and Turner syndrome, we have to look at a karyotype. But for things like developmental problems, we can look at sonograms, and that's where you get a picture of the baby. We can use blood tests to screen for proteins. This is going to be for genetic disorders. The sonograms are going to be used for physical disorders. Okay, if we can see physical problems in the picture, blood tests we can figure out just genetic disorders. And then we have two other called amniocentesis and chorionic villi sampling. In amniocentesis, what you do is you actually get a really big needle into the stomach of the woman, into the uterus. From there, you'll extract some cells, some fluid from the amniotic fluid, which is what the baby is sitting inside. That's what the water is that breaks. Okay? And from that, you can actually create your karyotype, which is the picture of the chromosomes that we just looked at. And we can identify Klinefelter's and Turner's and Down syndrome. So this is to study, chromosomal disorders. So the karyotype is for chromosome disorders only. Chromosome disorders. Then we also have chorionic villi sampling, and this is where you'll actually have to get tissue material from the uterus of the female, from the placenta, which is inside of the uterus. That's the sac that the baby is inside of, for a karyotype, but this is a lot more risky. And we'll talk about more of that in class later on. The last little section in our notes for Unit 3.4 is the idea of nature versus nurture. And this is which one has a greater effect on you. Does your genetics or your DNA have a greater effect on you? Or does your environment have a bigger effect on you? One of the best examples here is to use to test or to study identical twins. And we'll actually watch a video about twins in class. But take a minute to copy down these notes to think about what has that greater influence, your genetics or your environment. So what has the greater influence? This could be your DNA, your genetics, or maybe your home life, your environment. Think about how diet can have an effect and how environmental toxins can have an effect. So take a minute to pause, copy this down. And for the last thing you want to do is in your summary box, copy down this question: how can twins be an example of the effects of nature versus nurture? And answer it. Give me an example. Why do you think that they could be a good example of nature, your DNA, versus nurture, your environment? And that's all for today's notes.